Sorry about that. Welcome to Lecture 2, Part 1 of Lecture 2. So today we'll be looking at cell damage and deterministic effects. So this lecture I think is divided into 3 parts. This is the first part. So let's have a look at radiation toxicity. So it can be the biological effects of ionising radiation can be studied at different levels. So, for example, now let me get my pointer up. There we go, and let me get a better colour for you so it's easier to see. Good. Alright, so it can be studied at molecular level, so the DNA, proteins, etc. Subcellular, so the membranes, chromosomes, etc. At a cellular level, so there's the cell stops dividing or loses function. At the tissue level, so you get radiation syndromes, gastrointestinal effects, that sort of stuff, which we'll cover later. There can be a whole body effect, so death or cancer, or even a population effect. So if you irradiate a population, you can get genetic effects of radiation. So radiation, if you irradiate a biological material, you're going to get a random distribution of energy deposition in the cells. So a lot of the deposited energy is going to cause excitation and ionisation, which will sometimes be enough to break a chemical bond. So that is the main issue that we that affects, or the main biological effect is the actual chemical bond breaking. So the random distribution is responsible for the biological effects. So let's have a look at different phases. So if we go through a timeline of starting at 10 to the minus 15 seconds, you get like a physical phase. So this is where all the interactions take place. Then there's a chemical phase, which takes a fraction of a second up to maybe a second or so. So that will be the various chemicals being formed, and then there's a biological phase, which is the actual effect on the living organism itself. So here's some slides. This is the same slide as the previous one, but sort of maybe set out a little bit differently. So the physical phase is the absorption of radiation, the ionisation. Then the chemical phase is the direct and indirect molecular damage, which we covered a little bit last week. And then that's going to go on to the biological phase. So this is going to be observable biological effects like cell death, cancer genetics, etc., or cell mutation. Incorrect repair of cells. Whoops, sorry. So we've got a little bit of a flow chart here of what might happen. So there's absorption of the radiation by the material, then we get a molecular change down here. Then what can happen is the cell can repair normally and just survive as normal, or otherwise the cell can be damaged. If the cell is damaged, it can either die or it can cause malignant disease or genetic damage. If the cell dies, sometimes it's replaceable and will just be replaced. Sometimes it's not replaceable, and then if you have too many cells in an organ die, you'll get the organ stop function. So just going through, just doing a little bit of revision of last week. So the most important damage, radiation damage to a cell, is the damage to the DNA. So DNA damage in nature actually occurs quite commonly, and it can be from different agents. It doesn't have to be radiation, so it can be chemicals, temperature, light, or a range of other effects, or a range of other causes. The forms of radiation damage to cells can include damaging the nucleus and DNA, which is the main thing that we're interested in. Can also damage the cell membrane, which can rupture and let extracellular fluids enter the cell, and it can sort of basically drown the cell. It can, and the rupturing the cell can also allow leakage of nutrients from the cell. You can damage the mitochondria in the cell's cytoplasm, so it interrupts the cell's food supply, or you can damage the lysosomes as well to stop the cell or interferes with the cell's digestion. So the main thing we're interested in is DNA damage, and so it can damage the bases. So if you remember the DNA is like a ladder. So it's like a ladder, and it's got strands. The strands are like the outsides of the ladder, and then the little rungs are like the bases of the ladder. So you can get cross-links between the strands of DNA. You can get protein cross-links. You can get molecules inserted into different structures in the DNA. You can get disruption of the chemical bonds, but the main thing that we're interested in is the single and double strand breaks of the DNA. So breaking the actual outside or breaking across there. These strand breaks can be repaired, although the greater the damage, the less likely that the repair will be error-free. And then the outcome of incorrectly repaired DNA can be cell death or malignant disease or genetic damage. Let me get all of these up. Okay. So a double strand break in DNA requires break in both strands of the DNA about 2 nanometers apart, or within 2 nanometers. Some breaks can travel along a little bit, but it still requires for 2 ionisation events to occur within a few nanometers of each other. The number of single strand breaks and base damages per unit radiation is about 2 orders of magnitude larger than the number of double strand breaks. However, the number of double strand breaks correlates best with the cell kill. So it's the number of double strand breaks which is the most important. So the pattern of radiation damage is different for different damaging agents. So for example, ionising radiation as an agent produces a fairly large number of double-strand breaks, but cells have developed mechanisms to survive despite DNA damage. So you get repair mechanisms, and repair mechanisms are independent of the type of damage. So DNA can, if it's broken, it's broken, and then it can repair itself by whatever mechanism. DNA repair is a complex process which we won't go into very much at all. This is just basically background information for us. So there's some review articles there if you're really interested in it. But again, this is not